Okay, so in this video we're going to be going through transcription and translation, and it is important to remember that this is part of protein synthesis. So it's easy to get this confused with DNA replication, but it's important to remember this is protein synthesis. So if we're remembering that this transcription takes place first and that occurs in the nucleus is where we're going to start off. So if we're thinking about we're in the nucleus, and what we're going to happen first is similar to the start of DNA replication. DNA is going to unwind, and this is catalyzed by our, let me just make sure I've got this right, I think it is gyrase enzyme. Yeah, it is. So we've got the gyrase enzyme. Okay, and then we're going to have it unzipping. So again, unzip, got hydrogen bonds breaking. This is catalyzed by our helicase enzyme. Again, so we could get a mark for unwind, unzipping in the hydrogen bonds, gyrase and helicase. But it's also important to just make sure that you say where it's taking place, so in the nucleus, just to make it clearer. Okay. Third step then. So we are going to have complementary base pairing occurring. And we're going to have that occurring between the DNA strand and our mRNA that is going to have entered the nucleus. So unlike DNA replication then, here we're going to have thymine on the DNA strand. Will still be paired with adenine on the RNA, but there's no thymine in RNA. So if we have, I'll show you here. So if we have DNA, this strand, if we had A, T, C, G, and then we'd have our mRNA, we don't have any thymine on our mRNA. So what we're going to have here instead is uracil, but we would still have adenine, guanine, and cytosine, sorry. And then I'm still going to remember that we're going to have 3 hydrogen bonds between guanine and cytosine, so 3 again, and then we're going to have just 2 between A and U and T and A. It's always useful to make a note of that when you're answering the question. So then we're going to have, fourth step, the RNA polymerase. Remember, this isn't DNA polymerase because we're focusing on the RNA. And what that's going to do is going to form a phosphate sugar backbone. Just going to have it forming phosphate sugar backbone. Right. And then, so we've got mRNA from the complementary base pairing, we can see now, so this strand would be what we call a template strand. So I'm not sure, you can't really see that I wrote there, but template strand. And this, now our template strand is going to, hang on, let's move that up a bit, there we are, going to leave the nucleus because DNA is too large to leave the nucleus, so we're going to have mRNA leaving. Because our hydrogen bonds are going to break as it's going to leave the nucleus. So that is the transcription part done. Okay. So we've ended that there with the transcription. We've ended it with mRNA leaving the nucleus. Okay. So then what's going to happen, we're going to move on to translation now. Second stage of protein synthesis. So what's going to happen here is, so we've got the mRNA, it's left the nucleus, and it's going to bind to a ribosome. So it's a good thing to state that the mRNA has binded to the ribosome. I should use that word, bind. Okay. So another little detail that is important to add, it's something that, even like I said, if we forgot the stages and we just want to get something that's like another extra thing for another marking point, we can say that the start codon is always AUG. And an easy way to remember that is, well, this was an American way to do it, but school starts in August, which it doesn't, but you know what I mean, remember it like that. So if we've got our mRNA then, so we've got complementary, I'm trying to figure out how to word this, so complementary base pairing in fact, sorry, it's not complementary. So we've got on our mRNA, we've got some codons. Like the AUG is the first codon. So we've got our mRNA codons. And what's going to happen here is the tRNA, transfer RNA, is going to bring complementary anticodons. Okay? So if take the start codon, for example, AUG, this would be on the mRNA. We're then going to have on the tRNA, remembering because it's RNA, we don't have any thymine, so it'd be uracil, adenine, cytosine, and that would be the anticodon that the tRNA has brought to the ribosome. Right, and it's also important to remember that the tRNA will also bring a amino acid. So if we've got, let's try and draw this out for us. So we've got our this is mRNA, and then tRNA, this is in the ribosome. So tRNA is going to bring along, excuse the drawing, but 3 complementary codons, and it's also going to have an amino acid, and that's an amino acid there. So that's going to bind, we've got hydrogen bonding occurring. And then once this is binded, we're going to have another amino acid being brought, and the peptide bond is going to form between adjacent amino acids. So the main points there was that we've got mRNA with the codons, tRNA, bringing along complementary anticodons, and an amino acid where a peptide bond would form. So, and then we have, the polypeptide, which is what we're making, remember, because we're making a protein. The polypeptide is synthesized until a stop codon is reached. And at this point, the polypeptide is released. Yeah, and I think that's pretty much all we need to know for translation. Yeah. I suppose if we are struggling for marking points in the exam, you could say that 1 codon equals 1 amino acid. Okay, and that's protein synthesis for you. Thank you for watching.